Hello students, today we will study a new topic, structure of embryo sac cells and their functioning. Now here we will be selecting or studying the type which is typical embryo sac, that is normal type of embryo sac. We also call it monosporic, 8 nucleate or 7-celled or polygonic type of embryo sac. Please observe the diagram. You can see that we have an egg apparatus which comprises of 3 cells. Out of the 3 cells, 2 are synergid cells and 1 is egg cell. All are haploid. And we have got 3 antipodal cells towards the calyzal end. You can very well visualize and see in the diagram. And in the center, there is a central cell, actually the 2 polar nuclei which are haploid. All the cells in this particular embryo sac is haploid. And the central, we call it central cell. Actually, these are 2 polar nuclei. They do not fuse. They are called polar nuclei. If they fuse, they give rise to one diploid secondary nucleus. Or the 2 nuclei wait for the male gamete to fertilize, ultimately the triple fusion, and which will result in the primary triploid endosperm nucleus. First and foremost, we start with the synergids, the 2 cells of the egg apparatus. They lie towards the micropylar end. They are pointed, hooked, enveloping the egg or the female gamete on the 2 side. The cytoplasm of the synergid is strongly polarized. The calyzal region of the cell is occupied by one or many large or small vacuoles. You can see how they are enveloping the egg. Now let us talk about how the synergids behave, or what is the fate of the synergids. In embryo sacs with 2 synergids, one degenerates before the entry, the other one, called the persistent synergid, degenerates shortly after the embryo sac has received the pollen tube discharge. In Cordula australis, one of the synergids becomes swollen and hostorial in nature. Synergid hostoria have also been recorded in sedum and chordateria, a member of Poaceae. The hostoria arise as finger-like projections from the filiform apparatus and penetrate the embryo sac. Now let us study the functions of synergids. They play an important role in directing the pollen tube growth by secreting some chemotropically active substance. The degenerating synergids form the seat for pollen tube discharge into the embryo sac. Jensen 1965 suggested that the filiform apparatus may be aiding the synergids in absorption and transportation of materials into the embryo sac from the nucellus. Structure and function of synergids, the nature of metabolic activity, their differential behavior, entrance of pollen tube, and their role in opening of pollen tube needs to be studied in detail. We already know that these synergids are towards the micropylar end are provided with filiform apparatus, a prominent structure which is present at the micropylar end of each synergid. This apparatus is a mass of finger-like projections of the wall of into the cytoplasm. Structurally, each projection of the filiform apparatus is a core of tightly packed microfibrils made up of cellulose and closed by a non-fibrillar sheath. They are rich in polysaccharides. The form of filiform apparatus is variable. It may be spherical, as in Tauronia, or wedge-shaped between the tips of synergids, as in Pittonia and Helianthus. Egg apparatus. The 3 cells of the egg apparatus are arranged in a triangular fashion. The egg shows common walls with the 2 synergids and the central cell. It has a wall similar in structure and it extends to that of synergids. The wall is thicker in the micropylar region but becomes thinner towards the calyzal end. It is absent at the calyzal end in cotton and maize, etc. At the micropylar end, the lateral walls of the egg appear to join the central cell wall. Actually, the end cell wall of the egg cell is the wall of the central cell. The egg cell, the female gamete or oosphere, becomes highly polarized early in its development. The micropylar end of the cell is occupied by a large vacuole. Thus, the distribution of vacuole and cytoplasm in the egg cell is just opposite to that in the synergids. The egg may contain small or large amount of starch that is consumed during fertilization or early embryogeny. The egg cytoplasm is rich in ribosomes. The egg of Nicotiana rustica contains two strongly RNA-positive bodies that multiply and persist till the four-cell stage of proembryo. Towards the callosal end of the embryo sac, three antipodal cells are present. They are persistent with cytological features suggesting their role in nutrition of embryo sac. In Caltha palustris, they persist up to the octant stage of the proembryo. In grasses, they undergo a series of mitotic divisions leading to the formation of a large number of antipodal cells. The highest number of antipodal cells known is 300 in Sessa paniculata. Z maize has about 20 antipodal cells, each with 1 to 4 nuclei. Multinucleate condition of antipodal cells also occurs in Tagetes. Let us continue with antipodals. Antipodals are rich in ascorbic acid, oxidases, sulfhydryl compounds, starch, lipids, proteins. However, they show very low concentration of RNA and polysaccharides. Hostorial behavior of antipodal cells is known in many plants: Grandalia, Haplopeppus, Quinquecemelium, etc. Now let us study what functions they perform. Antipodals are nutritive in function. They nourish the embryo sac. The antipodal cells may also store large quantities of starch, lipids, and proteins, which are utilized by the developing endosperm and embryo. Antipodal cells produce and secrete substances that control the growth and development of endosperm. Being largest cell of the embryo sac with large central vacuole is the reservoir of sugars, amino acids, and inorganic salts. The nuclei of the central cell, called polar nuclei, are very large and each possess a conspicuous nucleus. Either they fuse before double fertilization to give rise to a diploid secondary nucleus, or the two wait for the male gamete and fuse and fuse during triple fusion to give rise to primary endosperm nucleus, which is triploid. Let us continue with the central cell. Unlike the egg cell, the cytoplasm of the central cell is rich in all cell organelles and appears to be the center of intense synthetic activity. In capsula, Shepherd's purse, the central cell possesses numerous spherosomes associated with glyoxysomes that would probably convert fat into sugar. There are numerous ribosomes usually present as single ribosomes or small polyribosomes. The wall of the central cell is highly variable from one part of the embryo sac to the other. The central cell is connected with egg, synergids, and the antipodals through cytoplasmic connections called plasmodesma. What function the central cell performs? The presence of cell wall projections, the micropylar or callosal region, shows that central cell draws nutrition from the surrounding nucellus or integuments. The hostorial extensions with parietal prolongation enter the ovular tissue. In addition to absorption, these extensions secrete enzymes that help in the digestion of the nuclear tissue. Last but not least, I would like to thank Rukma for nice drawings, and thank you to all for patiently listening. Like it, subscribe it, and share it. Thank you.